of insect symbiosis that has spread globally and led to major reductions in vector-borne diseases transmitted by humans. She is not just a force in transgenics, microscopy, cell biology, but she's also one of the world's experts in fly and mosquito reproductive systems, which I think you're going to be fascinated by today. As you take in RIM scholarship, you'll notice an extreme exemplary merit for organization, criticality, standard setting, and mentorship of the people and sciences she advances. And what stands out is that her leadership impact is done in a poised, calm, and always self-critical manner, the hallmark of science. She's the recipient of the 2022 Young Research in Biology from Venus International Science and Technology Awards, several research fellowships and oral presentations throughout her career, and a 2022 membership selection for the Genetic Society of America Early Career Leadership Program. So just a quick summary of her background. She obtained her bachelor's and her master's from Punjabi and lovely professional university, and it is lovely to have her. She postdoc at the University of Vienna in Austria, then to Portugal, and then thankfully joined us when we were at Vanderbilt University and made the trek with us from Vanderbilt to Penn State. And quite honestly, we would not be able to do what we're doing in the lab without her. She is the center of our research going on in the lab. So she's now a research professor making waves that you're going to hear about today. And I just can't say how thankful I am to get to work with her every day. And I'm just so inspired by the research that she does. So with that, Microbiome Center plaque. And I think this is amazing. If you guys want to come take a look at it, I'm really impressed by the craftsmanship that has gone into these. So please, everyone, welcome RIM.
Well, nobody find out. That's not my talk. Thank you, Sarah, for such a heartwarming introduction, and likewise, everything like you guys do for benefiting the lab and for making and heightening your people. It's amazing. So yeah, without any further ado, I would start my talk, and I'm really honored to represent Wolbachia Lab and the kind of work we are doing from the intracellular perspective of microbiome. Yes. So just to lightening the mood, how many of you have watched the movie Jaws? All of you, yes, that's what I imagined. So did I, and I was a kid when this movie was out, and I was so baffled with the concept of sharks. Like they are, they had a reputation of man-eating animals, and I was so afraid until I get to see this. This is the figure of the world's deadliest animals that kill humans per year. And guess where the sharks are? Right there at the bottom. So basically, sharks are responsible to kill only on an average of 10 human beings per year, which is still bad. But if you compare it with the rest of animals, and specifically when it comes to the world's deadliest animal that is the most dangerous for the human health is this tiny bug, which is mosquito, which is responsible to kill almost 1 billion of humans per year. So, and one of the example of this mosquito species, which is Aedes aegypti, which is primary vector of spreading a lot of viral diseases, just to name them, Zika, dengue, chikungunya, as we know, they are the pandemics, right? So it's really important to control the population of mosquitoes. And there are several strategies that we already use, like spraying insecticides, pesticides, but we all know over the time, they just develop anti-pesticide resistance and they are not working anymore. And moreover, they are not even eco-friendly for us to breathe in the air, which is so fumed with these pesticides. So WHO, CDC, they recommended to find a way, like a natural biological agent to control the mosquito population and save the human health. And that's where the Wolbachia comes into play. And this is the major player of my story. And this is the symbiotic bacteria. And I will tell you in a second why we are focusing on Wolbachia to control mosquito population. So just a little bit of background on Wolbachia. It was discovered in 1924. Two Harvard scientists, Marshall Herdig and salmon Wolbach, they were just ripping apart the ovaries of Culex ppn mosquitoes, and they found that all of them, they were having these rod-shaped bacteria inside. Back then, they didn't know which this bacteria is, what does it do, but they were fascinated because more and more insects they were looking into, they were always finding these bacteria. And they said that this type of organism is generally distributed throughout the group of arthropods. So since 1924 and cut to the chase, like today, we know that this bacteria is not only in mosquitoes, but it's infecting almost 50% of arthropod species and 50% of filarial nematodes. And when we talk about 50% of arthropods, that encompasses like millions and millions of just the insect species only. So basically, you can find Wolbachia everywhere. So, sorry, thanks. The picture on the right, it's a Drosophila ovary. Blue is the host cell that are labeled with DAPI. And in red, you can see Wolbachia. So I hope you can appreciate how infested this Drosophila ovary is with Wolbachia, where every single dot is one bacterial cell. So this is, it's an obligate intracellular bacteria. It only lives inside the host cells, residing in the cytoplasm. It is only vertically transmitted through the females. So males cannot transmit Wolbachia to next generation, only females can do that. And since it's mainly inhabiting the reproductive tissues like ovaries in females, testes in males, it has the ability to manipulate the host reproduction for its own benefit, which is basically where we'll be going in the next few minutes. So one of the most common host reproduction that can be manipulated by Wolbachia is called cytoplasmic incompatibility. It has other ways too, but the one which is the focus of my research and which I will be focusing on today is CI. What CI means is as the name suggests, is the incompatibility status between the cytoplasmic infection status of Wolbachia. So if in the gray there are individual mosquitoes without Wolbachia, and in green they are carrying Wolbachia. So if both males and females, they do not have Wolbachia, it doesn't matter, there's no incompatibility, the progeny is gonna live happily, right? There are mosquitoes, they will be also not having Wolbachia. Now there is an incompatibility going on here. Males have Wolbachia and the females do not. So this cross leads to the production of embryos that never see the day of light. They just die because of the incompatibility status. Now you would be thinking that there is an incompatibility here too. But as I said, the females have Wolbachia. So Wolbachia only needs female to transmit itself to the next generation. That's why the progeny lives and the Wolbachia gets transmitted, right? And if both have Wolbachia, again, nothing happens, the progeny lives happily. So in all these cases, I hope you can appreciate that the Wolbachia is giving advantage to the infected females in these two of these crosses over the uninfected ones to spread itself to the next generation, okay? And it's fascinating that the CI, this incompatibility between the mosquitoes was found in 1951, but the sole reason that Wolbachia is actually the driving CI was found in 1971. I just wanted to give an idea about the Wolbachia research timeline. And the Wolbachia's wonders do not just end there. It has another interesting capability that Wolbachia can protect the host against the antiviral. Wolbachia can provide antiviral resistance to its host. So this was the phenomena discovered back in 2008 by two independent groups, one in UK and one in Australia. They found that Drosophila melanogaster, which is a cosmopolitan Drosophila we all see on our trash cans, and they are infected with a strain which is called WMEL Wolbachia. So if the flies have Wolbachia, the natural pathogenic viruses that infect Drosophila, they are not able to replicate inside those flies. But if you take out Wolbachia WML, then this virus is able to replicate and also kill the host eventually. So presence of Wolbachia provide antiviral resistance. Now, when we are talking about Aedes aegypti as our primary candidate to target, notably, Aedes aegypti do not carry Wolbachia in nature. So the scientists across the world, they thought that what if we take this Wolbachia WML from the flies and inject it in the eggs of Aedes aegypti, and then see whether it will be able to block virus replication or not. Because Drosophila viruses do not affect us, but the mosquito viruses do. So they bring the Wolbachia into mosquito eggs, created mosquitoes carrying Wolbachia now, WML, and they tested against several virus transmission and the replicability, and found that this Wolbachia mosquitoes have now blocked their replication. And these mosquitoes are not able to transmit viral diseases to humans. So combining Wolbachia's ability to provide virus resistance and CI is now being used for vector control strategies. Two different ways. One is you can use CI if you release only Wolbachia infected males in the field where there is uninfected Aedes aegypti population. What will happen? The males will mate with uninfected females in the field and the population will crash because of CI, right? No eggs are going to hatch. So this strategy is called incompatible insect technique. And the second is population replacement strategy, which is being used in the field quite successfully now. What happens in this case, you release both males and females that are infected with Wolbachia in red here. So just a matter of time, they will sweep up the population of uninfected mosquitoes and replace that with infected ones. Why? Because Wolbachia gives advantage to the infected female. And they're going to mate with both uninfected and infected males in the field and propagate themselves. So eventually, over the time, your population will be replaced by infected ones. And those infected ones do not have the capacity to replicate viruses. So that's how this replacement method will crash down mosquito vector transmission capacity. So the strategies, the Wolbachia biocontrol strategy is now being backed by World Health Organization, Bill and Melinda Gates Foundation, and CDC to use this on a large scale. And actually there are nonprofit organizations like World Mosquito Program, Mosquito Mate, and Verily. These are the companies and organizations who are actually deploying Wolbachia infected mosquitoes in the field to achieve successful results. And as a matter of fact, they are now releasing mosquitoes in all these more than 10 countries where the dengue and Zika epidemics are super high. And interestingly, they have seen a range of 50% to 98% reduction of dengue itself. So in different countries, just a matter of release, in these many months post-release or these many, you will see reduction in the dengue cases. So Wolbachia and CI and the antiviral resistance strategies, they are being used very successfully in the field to control the mosquito population. Now we know that the field applicability, right, the field success, but it was not known what is the mechanism of CI, how Wolbachia is causing CI, and what are the impacts on the host reproductive biology it's causing that allows Wolbachia spread in the mosquito population. So this is the core of our the research domain that I'm working on. And I will tell you the various strategies, just like Sarah proposed. We are using genetics, cell molecular biology, and biochemistry skills to uncover the mechanism of CI, like how does Wolbachia cause CI? So first focusing on the genetics part, this was the work done back in 2015 in our lab. So what people, Bordenstein lab members, what they did, they used the comparative genomics, proteomics, and transcriptomic approach. So they used the Wolbachia strains that have the capability to induce CI, and then they compared their genome, proteome, and transcriptome to the ones that do not cause CI. And on that, long story short, they came up with two candidates that were only present in the CI causing strains. And these they call CI factors A and B, CIF-A and CIF-B. So to give you a zoom out view, this is the fly. It has Wolbachia in its reproductive tissues, and Wolbachia genome has a prophage now. And they found these two genes are lying around in the prophage genome, which is integrated in the Wolbachia genome. And these two genes, CIF-A and CIF-B, they are in the eukaryotic association module, meaning that they have the capability to interact with the eukaryotic host to do whatever it needs to do. So now they had the candidates and they wanted to know if these are actually causing CI or somehow associated with CI. So they tested that using a Gal4 UAS binary system. We use this system in Drosophila melanogaster a lot because it helps us to understand the ability to just dissect the role of one single gene versus the whole Wolbachia. And notably, Wolbachia is an unculturable bacterium. You cannot culture it outside the host. So that's why we are using the transgenic to study the role of the Wolbachia genes. So what happens in here, the Gal4 UAS system only works if it's together in one single fly. So Drosophila biologists, what they do, they have one fly that have a transgenic construct carrying Gal4, and they have another fly that carries UAS transcript. So what happens when you cross them, then they become together in the progeny. Gal4, which is a transcription activator protein, it activates UAS. UAS that lies upstream of the transgene that we want to study. It activates the expression of the transgene. And on top, you can drive the expression of this transgene in your tissue of interest. So you can attach Gal4 with the promoter where you want to express the gene. For example, reproductive tissues, because we are studying Wolbachia here. For that, we use nanos, which specifically express in reproductive tissues. Okay, so the way to test is, this is a kind of Punnett square where filled symbols are getting Wolbachia and unfilled are not getting Wolbachia. So this is the CI cross, males are infected, female uninfected, and this is the rescue cross, where filled females infected with Wolbachia rescues lethality. So on the x-axis, you have embryos that hatch out of the cross of these male and female. And every single dot is the number of embryos hatched from the eggs laid by one single female. So when there is Wolbachia infected male, uninfected female, CI occurs because not a lot of embryos hatching. There is 80% CI going on. And when there is female uninfected, it's a rescue. All of the embryos hatch normally. Now to test transgenics, CypA and CypB, what they did, they removed Wolbachia by antibiotic treatment, and they only used the transgenic expression of CypA and CypB males. Interestingly, none of them cause CI, only until they found that if you express both of them together in these males, they cause CI. So these genes, the expression of both genes in the male germline is essential to cause CI. And the next question is, what is the rescue mechanism? Like what is the genetic basis of rescue? So they expressed both CypA and CypB in the females now. And they showed that CypA alone is enough to rescue CI, CypB does not rescue, okay? So it's a 2-by-1 model where 2 genes are necessary to cause CI in males and 1 gene is necessary to rescue CI in females. Okay. Moving on to the second part, where I come in the picture, cell and molecular biology. So just to give you an overview, what we already knew from the cell biological basis. So when the fertilization happens, we know that CI is embryonic lethality, right? So when the fertilization happens, this is the first mitotic division when sperm fertilize the egg and then male and female pronucleus, they come together and then the first mitotic division starts. So both male and female chromosomes, they start separating on the anaphase, right? On 2 opposite poles. So here you see that both the maternal and paternal chromatin, they are separating nicely. The embryo is dividing nicely. They are perfect nuclear divisions going on. But in case of CI, what happens is the paternal chromatin is unable to disentangle to the opposite poles. So the maternal chromatin do, but the paternal does not. So this causes mitotic failure. The nuclei, they fail to divide. And even if they are able to divide, there is a chromatin bridging going on. So 2 nuclei are joined together. So this eventually leads to embryonic death as well. So with this knowledge in mind, we asked the question, we all know that here what is happening is a paternal chromatin, which is under the radar here. So we asked the question, how the sperm chromatin is being modified. What is the impact that CIF proteins do on the sperm chromatin, which eventually kills the embryos, right? So to answer that question, we use the mosquitoes from the World Mosquito Program, which are actually being released in the field. So we looked at the spermatogenesis process first. And this is the Aedes aegypti mosquitoes, and this is the testes magnified view. Spermatogenesis process in the insects is a multi-step process where on the apical tip you have stem cells, and when they divide and undergo mitosis, they become spermatocytes here. And these spermatocytes then grow and undergo meiotic divisions. Then they become leaf stages. And these then start elongating. You see that the sperm head and the tail, they are started forming here. And then after this spermatic differentiation, the nuclear volume of the sperm head, it starts even shrinking more because the sperm DNA is condensing here. So then they become needle shaped, and eventually upon maturation, the mature sperms go out and fertilize females. So we looked at CIF-A and CIF-B localization in the spermatogenesis process. So top panel is CIF-A. We designed the antibodies that target CIF-A protein. And bottom is merge of CIF-A and DAPI. And DAPI is used to label the host nuclei. So first stage, the stem cells, we see that CIF-A is co-localizing with DNA. And some of that is also in the cytoplasm. Moving on to spermatocytes, CIF-A in the cytoplasm. So it's shuffling between nucleus and cytoplasm. And in the leaf stage, it completely goes into the nucleus as it's overlapping with the DNA. Upon elongation, there is no overlap between DNA and CIF-A. It comes out of the cell again. And at the needle stage, we see that the nuclei are here and CIF-A is kind of trailing back on the tail, right? So CIF-A shuttles between nucleus and cytoplasm, and ultimately it comes out and stick on the tail of the sperm. Now looking at CIF-B, we see that in the initial stages in the stem cells, spermatocytes, CIF-B is cytoplasmic, and then it moves to the nucleus of the leaf stage. Almost like because CIF-A and B needs to express together. So we think that CIF-A, which is in the nucleus, it comes out, takes CIF-B and shuttles into the nucleus here. So it could be one of the hypotheses how they work together. So after CIF-B comes into the nucleus, it decorates itself over the sperm head. And in the needle stage again, it's decorating on the sperm head nuclei. And in the mature stages, when it comes out in here, it's also loading on the sperm head. So CIF-A comes out on the tail, but CIF-B sticks on the head. Now, after looking at the localization, we asked the question, now we know that they interact with the sperm chromatin, they are present there, but what are they doing to the sperm's process? So in the spermatogenesis process, the sperm chromatin integrity is a very essential process for the whole spermatogenesis process to go smooth and make a fertile, normal sperm. So what this transition means, these are the early stages, these are the late stages of sperm. In the early stages, the sperm nucleus is flooded with histones. They're bound with histones, right? So DNA is wrapped around them. And there comes a stage where the histone to protamine transition happens, meaning that histones, they start coming off and they go degraded. So now you see the DNA was wrapped around histones, but then histones are coming off. That means DNA is now fragile. It's vulnerable for DNA damage, right? So that's where the DNA damage and repair happens. Cell knows it's in situ phenomenon. So cell knows that I have to repair this DNA damage immediately when it happens. So this is in situ process. Histones degrade and then the protamines, they start accumulating on the sperm head. And they tightly coil around the sperm head to make it super tight and condensed form. And interestingly, this process of histone to protamine transition for a normal spermatogenesis is highly conserved from fruit flies, mosquitoes, amphibians, mouse to the humans. And in humans and mouse, it is known that if you abrupt this histone to protamine transition, it leads to embryonic defects, like the post-fertilization, miscarriage, or abortion happens. Okay, so we looked at what SIFs can do to this transition process. So this is the early canoe and late canoe stage where the histone to protamine transition occurs. Top panel is when there is no Wolbachia, and the bottom panel is when there is Wolbachia. And in purple, it's histones labeled with histone antibody. In blue, it's DNA merged with the histones. So in WML negative at this stage, when the histones need to come off, we see very less intensity of histones, suggesting that the normal process of histone removal is happening. But in MEL plus, a lot more histones are retaining than they should. And we quantify this phenomena by quantifying the intensity, and it's clearly more in WML plus. Then in the needle stage, can you see these dots? Okay, so histones are still retaining on the sperm head, while in WML negative, you don't have histones anymore. In the mature sperm, when they come out, still there are histones retaining, and in mature sperm in MEL negative, it's completely gone, just like normal process. So suggesting that WML plus, the SIFs in the WML plus males, they're definitely perturbing this histone removal process, and they are abnormally retaining histones here. And we quantify the puncta in the sperm that have histones in MEL plus and MEL negative. Now we looked at the protamine composition of the mature sperm, because histones are replaced by protamines, right? So what is happening to the protamines? And to do this assay, we use a stain. It is called CMA3 stain. This stain competes with protamines to bind to the same regions on the DNA where the protamines bind. So basically, if you have less protamines, you will have more CMA binding, more CMA intensity. And if you have more protamines, you will have less CMA binding, right? So it's an inverse way of reading the data. And this is what we see here in the sperm from MEL negative males. There is less CMA intensity, means more protamines. And in MEL plus, there is more CMA intensity, meaning less protamines. So obviously, SIFs in the Wolbachia infected males, they are leading to abnormal histone retention and reduced protamine deposition that affects chromatin integrity. And to discover and to verify basically if protamine deficiency is actually linking to CI or not, we use the help of Drosophila because you can do mutagenesis in there. So we used protamine deletion mutants. These flies, they do not have protamines. And minus means they don't have Wolbachia, plus means they have Wolbachia. And these are our MEL negative and MEL plus Drosophila controls. So what we see that if protamines, mutants, they have Wolbachia, and this is the wild type CI level, which is 40%, 60% CI going on. If you have protamine deletion, then the CI levels are enhanced. So basically, deletion of protamines can enhance CI levels. So this gives us not a direct correlation of protamine deficiency to CI, because in the uninfected one, there is no CI going on. There are still no protamines. But if there is protamine deficiency together with Wolbachia presence, then definitely CI is being accentuated, suggesting that Wolbachia and SIFs are interacting with the protamines to do phenotypic changes that associate with CI. Okay. So to summarize, cell biological findings, SIF A and B interact with sperm chromatin during myogenesis, and they impair sperm chromatin integrity by perturbing histone to protamine transition. Right. So moving on to the third aspect, which is the biochemistry. We know now that SIFs are present in the sperm, they interact with them, they modify them. But how? What is their biochemical properties that allow them to do so? I will take you back on this slide. And if you remember, there is a DNA damage, right, going on here. And in humans and mouse model, it's known that if this DNA damage doesn't get repaired on time, this leads to sperm infertility issues. So we asked that SIFs are definitely changing histone to protamine transition, but are they also manipulating DNA damage process? So for that, we first did in vitro enzymatic assays. We checked if SIFs can metabolize nucleic acids. So with using recombinant SIF A and SIF B, we incubated them in vitro with DNA substrate, single-stranded, double-stranded DNA, and single-stranded RNA. And in all of these cases, we find that in the absence of EDTA, CIF-A and B both cleave DNA substrates. And when it comes to RNA, it's only CIF-A that cleaves RNA substrate and not CIF-B. And our reactions controls are EDTA plus because EDTA is a chelating agent. It does not allow nuclease protein to go to its substrate and do the nuclease activity. So it quenches the nuclease action. So that's why we don't have any nuclease activity here. Has control. So this data suggests that CIFs definitely can cleave DNA and RNA, depending on which CIF we are talking. So now we wanted to know in situ what is happening on the sperm level. And this is a way to detect DNA damage in the sperm from the mammalian knowledge. It's called a tunnel assay. If the sperm DNA is damaged, if there are DNA breaks, then the enzyme, the DUTBs that come in the tunnel assay kit, they are going to integrate in these nicks and they glow. And the enzyme TDT catalyzes the reaction. So more glow means more DNA damage, less glow means less DNA damage. And we tested wild type Wolbachia male plus males versus negative DAPI staining host nuclei, sperm nuclei bundles here, and tunnel that detects DNA damage. So we see that in presence of Wolbachia, there is enhanced DNA damage at the stage where histone to protamine transition happens, and there is less DNA damage in the male negative males. We quantified this across a lot of testes that we examined. Maximum of the testes that showed WML plus that showed 100% sperm bundles with DNA damage versus WML negative where the damage was minimum. And we also tested this in the transgenic settings in the absence of Wolbachia. So if you express only CIF-A and B in the reproductive tissues, we see that the DNA damage is even enhanced versus negative control. And this DNA damage, which is happening in the sperm, we then tested what happens to the embryos, which get fertilized with the damaged DNA. We see that in the CI embryo, this is a stage where they are almost half developed in the middle. But if you see here, the nuclear division is not as uniform as it should be. And I see that the DNA damage is only happening in these nuclei, which are going to kill the embryo. And this embryo dies then. And this DNA damage is being detected using PH2AV antibody, which labels the histones. And these histones only get phosphorylated when the DNA damage is happening. Otherwise, they don't give a signal. So this is what we see in the rescue embryos. The embryo developed nicely. There is no DNA damage happening. So suggesting that CIFs cause DNA damage in the sperm that even persists in the CI embryos. So this is the summarized model of CI. In the testes, sperms are developed with DNA nicks and abnormal histone protamine structure that changes their integrity of the chromatin. And when this damaged DNA is ejaculated and fertilized the ovum, the egg of Drosophila, then in the normal setting, it will be repaired, but in the CI embryos, the repair system gets damaged. So this DNA damage leads to chromosomal abnormalities that cause CI embryonic death. Okay, so this was all about CI, and we also now want to ask what is happening from the rescue side. So if you remember, CIF-A alone in the females, it rescues CI. And we looked at mosquito ovaries because CIF-A expression in the females, where exactly during oogenesis CIF-A is localizing and what it is doing to host. So in mosquitoes, the oogenesis process is a two-step process. One is pre-vitellogenic, when mosquitoes have not taken the blood meal, where the ovaries look like this. It has the germarium, the germ cells, and then it has a primary egg chamber where the nurse cells and the oocyte is developing. And the nurse cells provide nourishment resources for the oocyte development. So, and then mosquitoes take the blood feed. After blood feeding, the germarium stays germarium, the primary egg chamber, it starts developing into yolk. So nurse cells become bigger, the yolk starts getting developed in the oocyte, and the follicle cell starts forming. And there becomes a secondary egg chamber. So in one blood feeding, mosquitoes can lay a lot of eggs that become from the primary egg chamber, and then the secondary undergo primary again from the second blood feed. Okay, so in the pre-vitellogenic stage, when there is no blood meal, we looked at CYP-A in green, and DAPI is labeling host nuclei in blue. So you see this is the magnified view of one of these varioles. CYP-A is in the germarium. It's co-localizing with the nucleus, but it's nowhere in the nurse cells. It's only in the germ cell nuclei, and some dots around the periphery of the follicular cells. However, when the mosquitoes take blood meal, CYP-A goes into the nuclei of the nurse cells and also sticks to the germarium. So this is interesting for us because CYP-A goes to the nurse cell nuclei only after blood feeding. And the nurse cells at this stage, they are synthesizing and transporting RNA and proteins, which are important for the egg development. So CYP-A, we think that is definitely interacting with the germ cell nuclei right from the beginning and nurse cell nuclei to modify the egg in some way that eventually rescues CI lethality. And we looked at the embryo. Is CYP-A interacting in the embryo? So these are the rescue embryos, and we see that CYP-A is directly interacting with the separating chromosomes and a 2-hour-old embryo, which is dividing nicely, all the nuclear divisions. So this suggests that CYP-A starts interacting with the host chromatin, maternal chromatin, right from the oogenesis and stays with the nuclear chromatin until the embryogenesis. So we still have the burning questions from the rescue side, which we are now working on. What is the indirect or direct mechanism of CYP-A-mediated rescue? Is it directly interacting? Is it modifying something in the maternal chromatin? Or when does that modification happen? Early in the oogenesis or later in the embryogenesis? And are the chromatin changes in the maternal side due to CYP-A are same or they are different than the maternal ones? So these are the kind of questions we are wondering about and we are working on in the future. With that, I would end my story, and I would like to acknowledge Seth and Sarah Bornstein Lab. They have been immensely helpful. And all of the collaborators that are labeled with the star here, and the members of Bornstein Lab that were involved in all the aspects of this work, from genetics, cell biology, and biochemistry. And of course, our funding agencies from Vanderbilt and now Penn State and NIH. And with that, I would really like to thank you guys for being so patient and hear about my story. So thank you.
Very much for that fascinating story, Ray. And yeah, we're happy to take any questions. We'd like to start with a question from a trainee. Okay, since I'm also a trainee, I'm gonna start with the question. So, well, you know, I know you're still looking into this, but like, so any speculations on how CYP-A might be mediating rescue? Like from your previous findings, it seems like it has like a nucleus activity.
Yeah.
So it kind of doesn't make sense to me. So I don't know if you can.
Yeah. So CYP-A is CYP-A has DNase as well as RNase activity. So we have now proven its RNase activity in situ as well. I haven't shown the data here, but we have going on in the background that CYP-A can cleave sperm RNA as well. But from the embryo side, we haven't checked that yet. So that could be a possibility that probably is chopping up some of the chromatin to probably, so you know how CIX maternal chromatin separates on the chromosome, but the paternal does not. So probably CYP-A is modifying maternal chromatin in a way that it syncs with the delayed timing of paternal chromatin. It also does some changes that delays the separation of maternal and sync with the paternal one, right? Far-fetched, but we don't know yet. So that's one of the hypotheses, that it could be, delaying the timing of the maternal to match with the paternal, so that they eventually separate on the same time. Or if there is direct mechanism, it could be helping the DNA repair system in the sperm, right? It could repair that damage, so there is no chromosomal abnormalities, and embryo is happily dividing from the rescue side. But yeah, what do you guys think? Yeah, give me some ideas. These are some of the ways that we are thinking rescue could be happening, but we still have some concrete plans for that.
Looking forward to see what you'll find. Yeah. All right. Any other questions? Yeah. I'm gonna start with Jordan. Yeah.
Sure. So I guess like a related question. like if you actually just look at the sequences of CYP-A and CYP-B, like what would they be predicted to be? Like, can you make any inferences from that information?
Yes, they have predicted domains. CYP-B has nucleus domains, which goes together with the CYP-B's nucleus activity. CYP-A has some predicted domains, but very uncharacterized activity so far. So CYP-A has DUF domain, which is a domain of unknown function, right? And there are several DUF, like 719 to 134. There are different families that are involved in the nucleus activity too. So we don't know our DUF domain in CYP-A can be related to that family or not. So that's what we're working on. Second, it has a transcription factor, which is at the C terminal. So also we are thinking that it could be, affecting the transcription. And most notably is the CYP-A on the N terminal has a nuclear localization signal. So we, on the other hand, we deleted the nuclear localization signal, and we see that the CYP-A is not able to go into the nucleus now. So that part is really important to go into the nucleus, do its job, what it has to do, right? So yeah, that's what we have for now.
Based on one of your figures, it would insinuate you have many different sequences of these proteins. Have you ever thought about looking at conservation, like in those different domains, to see perhaps what might be most important?
Yes. So if you align the B's, there is a conserved core of these nucleus domains. So there are, it's a long story. So there are various different types of CYPs, and they are being phylogenetically classified into type 1 to type 5. So type 1 CYP-Bs, they have 2 nucleus domains and 1 DUB domain, the deubiquitinating domain, which is now being proposed to stabilize CYP-B's activity. Then there are type 2 to type 4, which do not have the DUB domain. They have only the nucleus domains, and they still cause CI as well, right? So the functional characterization of these domains is happening now, and a lot of labs are now working on it. And hopefully by the next time I do the meeting, I will have more information like what exactly those domains are involved in the functional perspective of this. Yeah, awesome. Thank you.
So yes, I had a question too.
Yeah, somewhere in your introduction, you were showing the efficacy since the time of release, and
Yes.
it's release in Brazil kind of stood out because it wasn't as effective as in other places, and it didn't seem to correlate with time, or maybe.
The Rio de Janeiro.
Yeah.
Yes.
Do you have any explanation for this?
Yeah. So these are the way they do these field trials. They are called RCTs, randomized controlled trials. So you have to have government permissions, community acceptance, right? Whether they allow you to release these mosquitoes in their vicinity or not. Brazil specifically did not allow RCT. So that's one of the factors. Like it was a random trial, and random trials are most likely to fail in these settings. So that's what I know from talking to these people. Do you happen to know more, Sarah?
No, I know a lot of it is governmental, and then I noticed across the board, there's also shifts in temperature and humidity,
Yes.
right? That can account for just the mosquito survival.
Like altitude and how far they're the place where they release the mosquitoes, how far was it from the city, and from the like a river or stuff. So all that matters.
Question from Theo.
Yeah, I was just wondering, so in the context of, working with these different governments to do these releases, what are the concerns around this?
A lot. Yeah. Some of the community members, they have raised concerns that what if Wolbachia can mutate, right? What if this Wolbachia mutate in the nature and it's not now providing the protection against the virus as it should, or temperature? Wolbachia density is supposed to decline with temperature. High density Wolbachia doesn't grow well. So in the locations where heat waves are going on, climate change is happening, like there's so much tropical environment. So in those cases, these methods may not be as acceptable and successful as they should be. So people are concerned about those, but to respond to those concerns, various scientists in Australia, this World Mosquito Program, this is in Australia. So the people, they're actually considering testing different Wolbachia strains, for example, WALB, which infects albopictus mosquitoes. And those are highly resistant to high temperature. They still cause CI. It doesn't matter you raise mosquitoes at 30 or 37. So it's going on in the back end to resolve those concerns. But yes, it's a good question.
Laura had a question, yes?
I was just wondering a little bit about the evolutionary history of these CIF proteins, because they're microbial proteins, right, that then do something in the host, but I'm wondering what they do for the microbe or maybe how these coevolutionary relationships have come to be.
Yeah.
Has anybody, sort of been publishing on that or?
Yeah, our lab just started. So yeah, Imchen is working on understanding the relation between microbiome, like gut microbiota and Wolbachia. Is there interaction going on? Is that impacting CI or not? So probably in one of the next meetings, he can present his data, and it's really cool. We have some great insights that we would like to share in the future. So definitely, yes, there is interaction going on, and there is. Yeah, I'm really excited for the work. Sorry, I cannot speak much on that.
Yes.
Yes,
Did you say that those were in a prophage?
yes.
So, do you know what is the history of this phage? Can it excise? Can it kill the Wolbachia? Can it transmit?
Yeah, we think that this phage is lysogenic. No, lytic. I'm sorry. This phage is lytic. It can lyse the Wolbachia cell to release CIFs outside. And we have some preliminary data as well, where we see CIFs using immunobode labeling outside the Wolbachia cell. So we think they are secreted. But are they secreted because phage is lysing Wolbachia, or there is a secretion system by the bacteria which is shooting these proteins out? We don't know yet. But these are all the burning questions. I think Sarah may have more insights. She's a phage biologist.
That's actually my area.
Yeah.
But what's really fascinating about this particular phage is that we found this eukaryotic association module, and I named it that because a lot of the adjacent genes to the CIFs, if you look at the evolution, they're actually animal genes. So somehow this bacterial phage is through horizontal gene transfer exchanging genes with the animal host, not necessarily the Wolbachia host. And this phage is really novel. It dedicates up to half of its genome to these animal genes. So, and it's exchanging these genes all throughout Wolbachia, giving it all these really special characteristics. And I can't find this phage anywhere else except it's in the endosomiata holospora, which is in Paramecium, but it has the phage, but it doesn't have any of the animal genes. So
you think this is how the phage, how the seed gets out is by lysing the Wolbachia?
Yeah.
And then it interacts with the chromatin of the
Yes, exactly.
mosquito.
Yeah. And also it's very interesting because there's a lot of work on Wolbachia distribution, Wolbachia immunostaining in the sperm cell. Wolbachia only sticks to the sperm cell until the elongation stage, which is like middle. After that, there is no Wolbachia, and Wolbachia does not even transfer with the sperm. But CIFs, they still stay attached until the sperm gets mature. So it's definitely that they're secreted out of Wolbachia to attach to the sperm cells.
So there is also a syncing mechanism for the phages to become active.
yeah, we're not sure how the phages become active. Yeah, I can put them under heat, and of course that activates them. Which could be why we're seeing these differences in different temperatures. When you take these mosquitoes, put them into really hot temperatures, the phage and the CIF, I don't know.
That's so cool.
Crazy, yeah, this is awesome. Yeah.
It's like this whole tripartite, right?
yeah.
And then Imchen's now looking at all of this in relation to the microbiome in general.
Yes, oh,
Yeah.
beautiful system. And
I have another one.
Any-
There's,
yeah. I think we have time. Very good.
Any insight as to why Wolbachia cannot be cultivated? Do you know if there is some metabolic dependency between?
Yes.
Okay.
Yeah, it's a great question. And it has been $1 million question for the whole Wolbachia community. People have tried to culture Wolbachia. So actually Jason Raskin's lab in MC, back in 2006, they tried that. So you can take Wolbachia out of the cell and in the cell culture, you can maintain it, but only for like 6 to 10 days, and then it dies. So basically it can sustain itself, but it does not divide and grow, for replicating itself and make itself outside the host. It cannot sustain. Yes, it's definitely, there are different reasons why it cannot. Metabolic dependence is one of them. It accesses a lot of host genes to do its whatever it has to do. It needs host like actin filaments to move around, and multiply and spread to the next neighboring cells. And if you take the actin out, Wolbachia dies. So there are different levels of interaction that Wolbachia does with hosts to sustain it inside there.
Is it possible to do any genome editing on the Wolbachia itself, or is that like not possible yet?
People have tried CRISPR.
Yeah, no.
Failed. Yeah. We don't know the details because they never published the data, and I would really like to see the data, like what they did and what failed.
Is there any potential application beyond mosquito control, say like ticks or other vectors of viruses?
Yeah, definitely. Pests even, like Drosophilus zucchii is an insect pest in a way that it damages the food crop. So it's harmful for the economy. So Wolbachia has been advised for use as biocontrol, and also recently against some plant viruses. So there are viruses that affect the crops, right? So Wolbachia was tested against those viruses, and it also provide resistance against plant viruses, not just insect viruses. So it's broad application, yes. It just possibilities are endless, it seems. Yeah.
Any other questions? Happy. Thanks.
Yeah,
very much
thank you,
for
guys.
amazing story.